So today's speaker is Hélène Morlon. She got her BS in math from the ENS, the Ecole Normale Supérieure, and she is agrégée in math, which is quite impressive. She got her master's and her PhD in what sounds like sort of more lab types of biology, and then did a postdoc with Jessica Green and with Brendan Bohannon in Merced and Eugene. Worked with Josh Plotkin at the University of Pennsylvania, and then did one more year of postdoc at the University of California, Berkeley in the POTS lab. Your talk today is Understanding Biodiversity Patterns Using the Tree of Life. All right.
Okay, thank you very much. Good evening or afternoon or morning to everybody here, depending on where you are. Yeah, so Eric said a lot already about who I am. I'm a CNRS researcher in France at the Ecole Polytechnique. It's in Paris. And I'm going to be talking about large-scale biodiversity patterns, diversification, and the tree of life. Before I start, I would like to thank Eric for giving me this opportunity to talk at the Philo seminar. And usually when I have people in front of me, I tell them that I tend to speak fast and to raise their hands to make me slow down to ask questions. So here I can't see your hands, but please ask questions as Eric asked you. Okay, so as ecologists and evolutionary biologists, we are really interested in understanding global biodiversity patterns. We are interested in understanding why we see so much heterogeneity in the way that species richness is distributed both on the surface of the earth and on the tree of life. So we'd like to understand why we have on the planet some places that are hotspots of diversity, and why we have some clades where we have many species and others where there are only few. Obviously, species richness is very complicated and determined by many various ecological and evolutionary processes. But at the end of the day, species richness results in the balance between speciation and extinction events. So one of the main keys to understanding how biodiversity is generated and maintained is to find robust ways to estimate diversification rates and to try to understand how and why they vary across time, geographic regions, habitats, and taxonomic groups. This is what we do in my lab. And to do this, historically, people have relied a lot on fossil data, but what we do today is to rely on phylogenetic approaches. So we have bigger and bigger phylogenetic trees that are becoming available. The full phylogeny of mammals was made available in 2007, and in the last months, the full phylogeny of birds came out in Nature. So we have these big trees arriving, and that are most of the times made available. But in these trees, we're missing a lot of data. We're missing all the extinction events, and we're also missing all the speciation events that led to extinct lineages. So in order to estimate rates of speciation and extinction and understand what influence them from these data, we need to rely on statistical methods and on models of cladogenesis. So the very general approach to do this, to estimate diversification using this reconstructed phylogeny is very simple. We rely on stochastic birth-death models. The very simplest model is a model where we have one lineage, which is going to diversify with a given rate lambda, so it gives rise to new species, and go extinct with a given rate mu. And so the full process here is represented on the left. And what we see today is what we call the reconstructed phylogeny. We sample some of the extant species that are still, of some of the extant species, we reconstruct their phylogeny using molecular DNA data. And we have this object that is represented here on the right. And in order to estimate rates of speciation and extinction from this reconstructed phylogeny, what we do is to compute the likelihood of this reconstructed phylogeny under different scenarios of diversification. And then we can apply maximum likelihood or Bayesian approaches to fit the model to empirical data and get estimates for rates of diversification. So within this very general context, what I wanted to talk about today is about the role of ecological limits, boom and bust diversity dynamics, the paleoenvironment, geography, and population dynamics, and more precisely, how we can use phylogenetic data to ask questions about the relative role of all these processes. Okay, so let's start with the role of ecological limits and boom and bust diversity dynamics. The reason why it's important to understand whether ecological limits play a huge role in diversification is that whether diversity is constrained by ecological limits versus diversification rates leads to major differences in our approach to understanding biodiversity. If we have an idea that biodiversity is unbounded and accumulates through time, then we're going to try to explain present-day species richness mostly by the age and the diversification rates of clades. On the contrary, if we believe that there are strong constraints to ecological diversity, we're going to try to understand what the ecological constraints are and try to correlate species richness with current environmental variables. So what we did with my collaborators when I was at the University of Pennsylvania is to develop an approach that allows to test the hypothesis that we are under a scenario of equilibrium dynamics. So here on this figure, this is something that is a little different than what other people have done, which is to be looking at or testing and diversity-dependent dynamics, where we're going to have speciation and extinction rates that depend on the number of lineages at a given point in time. Here, what we're going to test is really whether we are at a point where species richness at reach sort of its carrying capacity, and we are at a scenario where there's still speciation, extinction, there's still a turnover in species, but the number of species remains more or less constant. And as I say, this is important because if we are under a scenario of equilibrium dynamics, then diversification rates don't really matter to explain species richness. What is going to matter is what fixes this sort of carrying capacity. To do this in the general framework I showed you before, we need to derive the likelihood of reconstructed phylogeny, and so that's what we did, requesting predictions from the coalescence process from population genetics. So we derived this likelihood, and then we applied it on a large set of phylogenies, 289 phylogenies spanning a wide range of taxonomic groups, using the repository that Mark picked to put together, and some phylogenies by Albert Phillimore and Trevor Price on birds. So here are the results. The graphs you see here, they show the number of phylogenies with a given support for a given model. And so basically, if the phylogenies fall on the right of the graph, it means that they support a scenario of expanding diversity, so not a scenario where we are at equilibrium dynamics. Whereas if the phylogenies fall on the left, they suggest a scenario of equilibrium dynamics. And so you see that there's a very large support for many phylogenies for a scenario where we are not in a situation of equilibrium dynamics. The graph on the bottom, though, shows that although we are not in a situation of equilibrium dynamics, rates are decaying through time. And so what this suggests is that we are not on this right side of the curve where we have equilibrium dynamics, but there is variation of speciation rate through time, and in particular declines in speciation rate through time that suggest that we might be on a trajectory of diversity declines. But still, this is showing that we can't try to understand diversity gradients by correlating species richness with current environmental variables, and that diversification rates play a major role in understanding diversity gradients.
So,
but now, yes, sure.
I mean, how many of these phylogenies were from disturbed environments?
From disturbed environments?
Well, from environments where maybe they're not in equilibrium, maybe humans have changed them recently or something like that.
That's a good question. So these phylogenies were from a big repository and coming from very different environments and very different groups. And whether they are disturbed by humans, this would be a process that would be happening at the very end of the history of the clade. So, for example, if there would be extirpation, like extinctions by human, this would happen at the very end of the history of the clade and would not be a signal that would be detected in a molecular phylogenies on that many million years. So really what we're looking at is more diversity trajectory over a million years until today than that's something that could not be at saturation because it has been disturbed by humans.
Okay, thanks.
Okay. Okay, so here the two scenarios that we've been considering and that are the focus of current phylogenetic approach to diversifications are a scenario of unbounded diversity dynamics and of ecological limits. But when we think about what we know from the fossil record and also what seems natural in terms of biological processes, whereby we're going to have clades that diversify well under a given environmental background, but then environment is going to change or another clade is arising that is better competitor and so on, we expect that there's more that diversity dynamics is more something where we have increases in species richness and then declines in species richness has different clades outcompete the clades that are present at a given time. So yeah, so as I said, this is seen in the fossil record quite a lot, and in particular, it's a pattern that has been shown for cetaceans. Here, that's what you see on the left from a paper by Cantor and Marshall, where we see for the cetaceans an increase in diversity until 10 million years ago, and then a decline starting 10 million years ago. So these patterns of diversity dynamics are known from the fossil record, but they are typically not detected in molecular phylogenies. In this example of the cetacean, so Cantor and Marshall really made this comparison between the diversity trajectories we infer from the fossil record and the one we infer from phylogenies, and with phylogenetic inference here, that's what you see in the table, the net diversification rate estimated at present is always positive, meaning that we have an increase in diversity towards the present, whereas we know from the fossil record that diversity has been declining. So what we did in this paper with Todd Parsons and Joshua Plotkin from the University of Pennsylvania was to derive the likelihood that correspond to this particular birth-death process, a birth-death process where the speciation rate can vary over time and lineages, the extinction rate can vary over time and lineages, and the extinction rate can exceed the speciation rate, which is not something that is a big deal to do to derive the likelihood expression, but hadn't really been considered in previous implementations of a process. and we can allow for partial sampling, whereby only a given fraction of the extant species are sampled. What we first did was to test whether it is at all possible to infer declines in diversity if they happen. So we simulated scenarios where the extinction rate is held constant and the speciation rate increases through time, such that at the beginning of the history of a clade, we have an increase in diversity, and then the extinction rate becomes larger than the speciation rate and diversity declines. And we tested whether it was possible to estimate the net diversification rate at present under this scenario, and in particular whether it was possible to find a negative net diversification rate at present when they're actually negative. So what you see here is in the x-axis the simulated net diversification rate, and on the y-axis the inferred net diversification rate at present, and you see that at least we obtain unbiased parameter estimates, even though they have large confidence intervals. And most importantly, when we have negative net diversification rate at present, then negative net diversification rate at present can be inferred from the phylogenies, showing that at least on simulated phylogenies, there's no problem to estimate declines in diversity. Now, when we move to empirical phylogenies, if we don't take into account the fact that rates vary across different clades, we're going to infer very low extinction rates, what is usually seen when we do phylogenetic inference. But when we take into account the fact that there are shifts in diversification rates, and here these shifts were supported by the phylogeny, when we tested for models with different rates for different clades, when we take this variation into account, then we can find diversity in dynamics that look like what the fossil record look like. So where we have first an increase in species richness and then a decline in species richness. To compare directly with the fossil record, we can sum up these different diversity trajectories that are shown here on the right. So we have a trajectory of species richness over time. We see that for recent rich clades, we see the pattern that we see most of the time when we fit models to phylogenies, a pattern of diversity increase. But for when we isolate these clades and consider different rates for these clades, then we see patterns of boom and bust diversity. And when we want to compare directly with the fossil record, we can sum up these diversity trajectories, and that's what you see here in the black curve, and the red curves are the confidence intervals for species richness for the fossil data. And so we see a pretty good agreement between the phylogenetic inference and what we know from the fossil record. So what this is showing is that it is possible, I'm not saying it's easy, but it's possible to infer boom and bust diversity dynamics using molecular phylogenies. And this is important because such dynamics are likely to be the rule for most clades of what diversity dynamics actually is neither unbounded or reaching an ecological limit, but wakes and wine has, as we see in the fossil record. So far, I've been talking about estimating how rates vary through time, but not much about why. And so that's a little bit of what I'm going to be doing here. And in particular, at the beginning of the talk, I said that we have many ecological evolutionary processes acting to affect speciation and extinction rates, many abiotic factors, many biotic factors. So far, we don't have good phylogenetic methods to test for the impact of biotic interactions on diversification rates, but we are starting to develop approaches to test the effect of abiotic factors on diversification rates. So first, what are the abiotic factors that we think might have a big influence on speciation and extinction rates? Well, first, climate has been proposed as a major driver of diversification. First, based on the fact that we see this pretty good matching between, for example, surface temperature distributions and diversity gradients, or also based on some theoretical arguments, such as those coming from the metabolic theory of ecology, where we think that there is an effect of temperature on energetics, on mutation rates, rates of genetic divergence, and finally speciation. So there are these arguments plus some direct observation of fossil data and comparison with temperature trends that tend to show that there might be a correlation between levels of species richness and temperature values. Okay, aside from climatic variables, concentration in carbon dioxide in the atmosphere might have major impacts on diversity dynamics. This also is based on observation from the fossil record. And the third factor that might be really important is sea level change. So we know that sea level has changed a lot in Earth history, and this obviously changes the amount of habitat available to species, and so probably extinction. When we have less habitat, there's going to be more extinction. When we have more habitat, there's going to be more speciation, and so on. So what we did with my postdoc, Fabien Condamine, is to think about how to directly test the effect of abiotic factors on diversity dynamics using paleoenvironmental and phylogenetic data. And this is something that is going to come out soon in ecology letters as part of a bigger paper treating of other things, but just it gives an idea on how to go about testing the effect of paleoenvironmental data on diversification rates. And still with the same principle, we derive this time likelihood corresponding to a birth death process in which the speciation rates can still vary over time, lineages, but also this time with a measured environmental variable. So we think that we might have estimates of temperature through time or of sea level through time, and we can incorporate this data in this process, and so we keep all of the possibilities that we had in the previous expression that I showed before with extinction rate that can exceed speciation rate. We can sample only a fraction of the extant species, and we can derive just pretty straightforward from the expression that is given in the earlier PNAS paper to derive a likelihood expression for this process where we're going to have rates of diversification that can depend on a measured environmental variable. So we're starting now to apply this method to a wide range of molecular phylogenies sampled throughout the tree of life, and again having some endotherm species, some ectotherm species sampled in the tropics, in the temperate regions, and so on. And we're using the paleoclimatic records from the Cenozoic given by Zakos, and just applying this the likelihood expression to all these phylogenies. So at the moment, we really have only preliminary results, and we only applied the method with a dependence of the speciation rate on temperature, which is given by the metabolic theory of biodiversity. So it's this form that you see here. And basically what this allows to do here with this example is if we infer positive alpha values, then this indicates that we have a negative relationship between temperature and speciation rate, whereas if we infer negative alpha value, then this indicates a positive relationship between temperature and speciation rate. So this allows to test whether there is an effect of the abiotic environment on diversification rates and what this effect is. So really preliminary results applied on a couple of clades. We have seen cases where this parameter alpha is inferred to be negative, so we have a positive effect of temperature on speciation rates, but also cases where we see the overtrend. So this is work to come, and we're excited to see, what the general patterns are. But basically, what this is saying is that we can test if we have phylogenetic data and measured paleoenvironmental data, then we can test the effect of the abiotic environment on diversification rates.
So, just out of curiosity,
Yes.
how do you test that it's not zero?
So that would be by comparing this model with dependency on temperature to a model where there's no dependency on temperature, and this is all comparable. Doing likelihood ratio tests if they are nested, doing AICs if they are not nested. All the likelihoods are comparable. I mean, we basically obtain the likelihood of the same data. So this is all comparable. It's all in a likelihood framework where all these models are all comparable, so we can really test the support for a model where temperature has an influence to a model where it hasn't. And so far we have, at least we have found quite several clades for which we see a significant effect of the abiotic environment, here temperature, on speciation rate. Does this answer the question?
Perfect.
Okay, thank you. Okay, so now what about geography? At the beginning, I showed maps of species richness, and so that's what we're trying to understand, but I haven't spoke a lot about geography yet. And when we see the patterns of latitudinal gradient, for example, across different groups, across, in mammifers and amphibians, in birds, and so on, we tend to think that there has to be also a latitudinal diversity gradient in speciation and extinction rate. So we tend to think that speciation is higher in the tropics and extinction is lower in the tropics. But this is not actually that obvious, and particularly because the Earth was mostly tropical around 30 to 40 million years ago, and only became or the temperate expanded around this time. So we have a lot of lineages that are thought to be tropical, and this combined with the difficulty for tropical lineages to expand to temperate regions where they're going to have to face cold temperatures and so on. There can be patterns in latitudinal gradient in species richness without any difference in the rates of speciation and extinction across latitudes. So this is the first reason why. So it's known as the tropical niche conservatism hypothesis, where we don't need to have any effect of the being tropical or temperate to have a striking gradient in species richness. And the other process that could explain a latitudinal diversity gradient without any effect on diversification rates is this hypothesis of ecological limits, where richness wouldn't be determined by diversification rates, but rather by the amount of species that we can pack in a given region. So maybe we can pack more species in the tropics than in temperate regions because there might be more habitats, or because of environmental variables, climatic variables, and so on. So that's what we wanted to test using the latitudinal diversity gradient in mammals. And so we asked this exact question: is there a latitudinal gradient in speciation and or extinction rates? This is what Vir and Schlotter did in 2007 on both birds and mammals. And what they found is that there is indeed a latitudinal gradient in speciation and extinction rates, but it's not as we would expect it to be. So we would expect speciation to be faster, so speciation rate to be higher in the tropics. And they in fact found that speciation rates were higher at higher latitudes. Second study, more recent, in 2012, on the global phylogeny of mammals, where they used models of diversification to fit and estimate rates of diversification for individual clades, and correlate this with mean latitudes at which current species occur. And they didn't find any correlation between diversification rates and latitude. So we asked this question again, using this time the global phylogeny of mammals, and not splitting the tree into small individual clades, but keeping the whole phylogeny of mammals. And we got latitudinal information from the Pantheria database. And then we this time didn't develop any method, but just applied something that was available on this data. So we used this GOC model, which is the following. Here we have A and B that represent two different geographic regions. And here, because we're talking about the latitudinal diversity gradient, I'm directly going to be talking about A for a tropical region, B for temperate regions, and A and B are species that occur in both biomes. So this model is the following. For species who are present in only one biome, let's say the tropical biome, the species can go extinct, or they can speciate in this biome, or they can expand their range and become a widespread species present in both the tropics and the tropical and the temperate biome. Same thing for species that are present only in the temperate biome, and for species that are widespread, so present in the two biomes, they can either go extinct locally, so go extinct in only one of the two biomes and become again either a tropical or temperate species. Or they can, there can be a speciation event, and the new species can occur in only one of the biomes. That would be one more species added to the tropical biome. Or finally, the widespread species can speciate by biome divergence, and so that would give one species present in the tropical biome and one species present in the temperate biome. And still the same principle. The likelihood of phylogenies associated with geographic location under this process can be computed. It's not an analytical expression, but it can be computed numerically, and so we can fit this model to the data, to the combined data, the combined phylogenetic and species location data. So that's what we obtained when we fit this model to the global phylogeny. We find a higher, this time the pattern that would be expected from the latitudinal diversity gradient standpoint, higher speciation rate in the tropics, a lower extinction rate in the tropics, which result in a much higher net diversification rate in the tropics. So this is for the mammals seen as a whole, but as we know that there must be a wide variation in diversification rates across groups. We also applied this model to orders within the mammalian phylogeny. And I think I forgot to say that this is work that is done by my PhD student, Jonathan Rollin, just came to my mind now. And so when we apply these models to the 8 richest orders, we find pretty consistent patterns with always higher speciation rate in the tropics, lower extinction rate in the tropics, except for carnivora, where we see the pattern that Virenschurter observed, where we have lower speciation rate in the tropics. And in some groups where the difference is not significant, but otherwise it's consistent. Oh, and also except for the lagomorpha, which have an inverse diversity gradient, and where we see an inverse pattern where we have lower extinction rates in temperate regions where diversity is the highest. So, the patterns we see for the diversification rates are really consistent with the patterns we see for the latitudinal gradient, meaning that where we see high species richness, we see high speciation rates and low extinction rates. So what this study suggests is that faster speciation and reduced extinction explain in large part the latitudinal diversity gradient that we see in mammals, meaning again that diversification rates really play a major role in understanding diversity gradients, contrary to what we might think if we're thinking about the fact that species richness is constrained by ecological limits, and that we try to understand diversity gradients by directly correlating species richness with environmental variables. So I'm coming to the last part, which means that I must have spoken pretty fast. I hope you're all doing okay here. But that will leave plenty of time for questions, so and I can slow down then, that's good. So last part, this is about understanding the role of population dynamics in macroevolution and in diversity dynamics. And more than that, it's really something which goal is to start putting together individual-based model for macroevolution. Well, this might not be the best model yet, but right now what we're doing, all the models I've shown are models that are constructed at the lineage level. So we're using the lineage at the unit of the diversification, and so speciation is really abstract, is not modeled by any microscopic process. And for extinction, it's the same. Extinction doesn't come from the fact that population size arrives to 0. It just comes from the fact that we put this extinction rate, which is very abstract and hasn't got any process model behind it. So that's what we're starting to do with my collaborator here, Amaury Lambert, and the student Marc Manceau. And to do this, we based on the neutral theory, on Hubbell's neutral theory of biodiversity, because it is at the moment the only model which is a little tractable and which allows to go from individual processes, birth and death of individuals and speciation mutations happening on individuals, to macroevolutionary processes where we have speciation and where we can potentially predict phylogenies. So phylogenies under the neutral theory of biodiversity have been investigated mostly by simulations, and what people have found is that phylogenies that arise from the neutral theory of biodiversity have a realistic imbalance, which is not the case when we look at birth-death models, lineage-based birth-death models with homogeneous rates across lineages. But in the case of a neutral theory, we obtain realistic tree imbalance, but unrealistic branch length patterns. So here what you see on the horizontal line are gamma values, which show the repartition of nodes in the phylogeny for empirical trees, and the free data points come from arise from the neutral model, from phylogenies simulated under the neutral model, and we see that gamma values simulated under this process are always much higher than what we see on empirical phylogenies. Meaning that the neutral model has in its basic form, as implemented now, is not able to reproduce phylogenies with realistic branch length patterns. So to move forward here, we relaxed two hypotheses of a neutral theory of biodiversity. The first one is the fixed meta-community size. So instead of having now this zero-sum assumption of a fixed number of individuals in the community, we have a total meta-community size that arise from a birth-death process at the level of individuals. So individuals are neutral in their demographic parameters. So they all have same birth and death rates, and this generates the process that you see on the left here, so genealogies of individuals. And if we only consider the individuals that survived to the present and their ancestor, we obtain this reconstructed genealogy that you see on the right. Now, to be able to have speciation events in this process, what we have is random mutations arriving on the genealogy as a Poisson process. So we just throw mutations on the genealogy, and then in the case of a point mutation model, first envisioned by Hubbell, its mutation event would create a new species. But here, this is not what happened. The mutation event is just a mutation event, and we're going to have a speciation. We're going to decide whether there's a speciation event when all the individuals from the two descendant populations from the speciation event are separated from all the individuals from the other population by at least one mutation event, meaning that they are all genetically differentiated. So this is the idea that speciation happens by genetic differentiation. And so that here on the left, you see this process of mutation and speciation happening, and on the right, you see the reconstructed phylogeny that it results in. So what we were able to do with this model was to be able to simulate it efficiently. And so we were able to look at whether it had realistic branch length and balance patterns. So what you see on the right are results for beta statistics. So okay, the first one on the left gave a gamma statistic, and the one on the right, the beta statistics. So on gamma, we're looking at branch lengths. The point on the far most right are real phylogenies, and the points on the left are simulations under different parameter values. But basically, by changing the mutation rate or the birth rate and so on, we can obtain phylogenies which branch length patterns are within the range of what we observed for empirical phylogenies, and same thing for imbalance. So we can find here parameters for this model where we reach where we are able to produce phylogenies that are realistic both in terms of branch length and imbalance patterns. And the second thing that we're able to do was to find an efficient way to compute the likelihood of the reconstructed phylogeny. Again, this is not an analytical expression. It's an algorithm that we can together that allows to compute the likelihood of a phylogeny and thus to infer the parameters of the process and to test different scenarios. So this is, I think, one of the first microscopic models that produces phylogeny, and where we're able to compute the likelihood of phylogenies arising from a microscopic model. We see that this model, which is completely neutral in terms of the demography of individuals, can explain the shape of phylogenetic trees, both branch lengths and imbalance, which doesn't mean that the world is neutral, but that these models are really well designed, are appropriate at least to start building individual-based model for macroevolution, which I think is important if we want to start incorporating more ecological and evolutionary processes into these macroevolutionary models. Okay, so to conclude, there was several examples of how we can use molecular phylogenies to understand diversification patterns and the ecological and evolutionary processes that generate and maintain biodiversity. The developments that are really needed are all these developments that incorporate microevolutionary processes, species interactions. We don't know how to do that. I already mentioned, but we start being able to test the effect of the abiotic environment on diversification patterns. As far as species interactions, we're not able to do much. And there's also a big thing that is not done at all at the moment is to develop approaches that are more readily applicable to the microbial world. And when we know that microbes represent most of the diversity on Earth, this is something that I think we really need to start putting together, is approaches that are more appropriate to study patterns in macro groups of microorganisms. And with this, I would like to thank all my collaborators, students, and to thank Eric again for this opportunity. Thank you, Eric.